Good morning everybody. Today we will be talking about fabricating scaffolds for tissue engineering applications. We have looked at some of the materials which can be used. We have looked at the natural polymers and synthetic polymers. So, we will also be talking about other materials. However, today we are going to talk about how scaffolds can be fabricated. So, as you all know from the previous lectures, we want to prepare a scaffold which can actually mimic the extracellular matrix. So, the extracellular matrix supports cells to adhere, grow, and it also provides cues and signals which help in the cells to perform the way they do. So, this helps in making sure that the cells integrate to form a tissue. So, when we are engineering this tissue, it is crucial that we prepare a scaffold which can mimic the functionalities of the extracellular matrix. So, let us try and recollect what are all the properties that we look for. First thing is it needs to be biocompatible. So, that is decided based on the material you choose. So, that will not be a part of the fabrication strategy itself. If we are looking at what parameters have to be brought into fabrication, then we have to see what else are there. One of the things which is crucial is the surface area to volume ratio. If you have a high surface area to volume ratio, you can load more cells, which means there will be higher cell density and there is a higher chance of the tissue functioning as it should. So, creating tissues which have this kind of a high surface area to volume ratio would be very effective. Another factor is pore interconnectivity. So, when you create pores in a scaffold, you need to have them connected to each other. Why is this crucial? When you have good pore interconnectivity, you will have better cell infiltration, which means the cells can actually go inside the pores and populate the tissue to form a living tissue. So, creating an interconnected pore would be advantageous. This can also help in formation of blood vessels. So, this way you would have sustained nutrient supply and toxin removal. So, this is an important factor which needs to be considered as well. Other than this, you also need to make sure that the material is fabricated into the shape and properties which fit the tissue which you are looking for. So, taking all these into consideration, you have to design strategies which can help in fabricating scaffolds. So, today we will talk about some of the more commonly used strategies for fabricating tissue engineering scaffolds. These are some of the strategies which we will be talking about today. The first one is called as a leaching method. So, this is a broad category within which there are multiple strategies which can be used for fabricating scaffolds. So, the three strategies which are commonly employed are solvent casting and salt leaching, or ice particle leaching, gas foaming and salt leaching. So, what is done here is a scaffold is prepared and gas forming salt which is leached out to create porous structure. So, we will see how it is done and how the scaffold would look like if this strategy is used. So, the next type of scaffold fabrication strategy is using microspheres. So, you can create microspheres using different strategies. It could be just a simple thing like preparing alginate beads, so which you might have done as part of one of your undergraduate lab programs, or you can create macroporous beads using different strategies, and you can also have these beads aggregate to form a scaffold creating a porous structure. So, these are different strategies which have been employed to create a scaffold using microspheres of different polymers. It could either be synthetic or natural. The next strategy is called as a phase separation strategy. So, here you have two techniques. One is the freeze drying method and there is also a thermally induced phase separation method. So, by creating a phase separation, you can actually create porous structures, and this also will provide good interconnected pores. So, we will see more about freeze drying method and see how exactly the scaffolds would look like and what is the principle behind the freeze drying method. So, you also have fiber spinning strategies. So, this is where you have different textile based fabrication methods which are employed in tissue engineering. So, nanofiber electrospinning, microfiber wet spinning and non-woven polymer fibers are all some of the commonly used methods for preparing fibrous scaffolds. In many of the extracellular matrix you have a fibrous structure. So, creating a fibrous structure using these kinds of spinning techniques can actually mimic ECM very effectively. In case of electrospinning, where you create nanofibers, you would also have a significantly large surface area to volume ratio, which can help in cell adhesion and proliferation. So, we will talk about electrospinning strategy and look at how those scaffolds will look like as well. So, next strategy is called as decellularized matrix. So, this is one of the one of the growing strategies which is being looked at. So, where you actually take the extracellular matrix from the native tissue. This could be either from a human donor or also from non-human sources from other species. So, what you do here is you remove the cells and all the cell based components. So, this is done through different methods. It could be using some chemical treatment or physical treatment, followed by complete removal of all the toxins. So, this way you create a matrix which resembles the native tissue. So, here the major challenge is actually to remove the cells and its components without damaging the integrity of the extracellular matrix. So, if you can do that, maintain the mechanical properties, physical, chemical and biological properties, then you would have an extracellular matrix which probably has all the desired functionality, and thereby it will be able to provide all the biological cues as well. So, this would be an ideal strategy to look at. However, it is a major challenge to make sure that the properties of the scaffold is not lost during the decellularization process. So, we will not be talking about this today, but you can read about this on many of the papers which are readily available. So, the last and the up and coming strategy which people are looking at is 3D printing. So, 3D printing is a novel technology where people are looking at how scaffolds can be prepared by bioprinting the scaffolds. So, here an ink is used. So, the ink will basically contain the material which you want the scaffold to be formed with, and this can be used for fabricating scaffolds based on the design we need using AutoCAD and other CAD based software. So, here you can also load cells along with them and print the 3D structure for the tissues. So, we will be talking about this in a future lecture. So, there is also another strategy which is called as self assembly. So, that will also be discussed in detail in a later lecture. So, now let us look at some of the more traditional strategies which are the leaching methods, where we will be talking about the solvent casting and salt leaching and also the gas foaming and salt leaching method. So, we will first start discussing this. Let us first talk about solvent casting and salt leaching method. So, this is an example of how you would use this method to prepare a scaffold. So, in the protocol given, you have three things. First is a solvent which can dissolve the polymer and the salt which you are trying to use for this method. So, the polymer chosen here is PLGA and the salt which is used is just NaCl or sodium chloride. So, these three are poured into one vessel and they are mixed to create a homogeneous mixture. This homogeneous mixture is then poured into a silicon mold where you keep it and evaporate the solvent. So, once the solvent is evaporated, you now have a solid structure, so which will have the shape as the silicon mold. So, here they have shown a disc like mold. So, you would end up with disc like scaffolds. So, now this scaffold which you have contains the polymer and the salt. The salt, because it was a homogeneous mixture while it was in solution, the salt crystals would have formed in different parts of the polymer scaffold. So, now the next step is to leach this salt. So, this is done using DI water. So, you place the salt polymer scaffold into the DI water and mix it for 48 hours. By doing this, the salt will actually get dissolved out, and you will end up with a disc shaped scaffold which is only the polymer. So, now this can be kept in a vacuum oven after freeze drying to remove all the leftover water. So, thereby you now have a scaffold which is prepared through solvent casting and salt leaching. So, how does this create a porous matrix? So, the salt which was present would have crystallized and formed salt crystals in the polymer mixture, in the polymer blend. So, this salt, when it dissolves out or gets leached out, the area occupied, the volume occupied by these salt crystals will now become pores. So, these porous structures are used for cell attachment and culture. So, these kinds of cells scaffolds would look like this under a scanning electron microscope. So, what you see here are images of salt leached scaffolds. So, you can see nice structures which are basically the regions where the salt crystals were occupying. So, when you look at the SEM images, you would also see that there are some areas which are dark black while some are grey, indicating that there is a 3D structure and there are crystals which have been formed all over, creating such pores. However, one of the limitations you would see is in many of these pores there is very poor interconnectivity. For example, you look at this particular pore. So, this pore actually is fully covered. You actually have a region where cells can go and attach, but they cannot penetrate into the scaffold if they go into this pore. So, this would mean the interconnectivity might not be excellent when you are talking about solvent casting and salt leaching method. To overcome this limitation, people have looked at gas forming and salt leaching method. So, this also uses very similar principle. However, instead of using a simple NaCl kind of salt, you use a salt which can cause gas to be generated. So, here what they have looked at is here is an example. So, where they have prepared a polymer gel using a non-solvent precipitation method, and the polymer gel paste along with the ammonium bicarbonate salt particles is mixed and you pour it into a Teflon mold to create a semi solidified polymer salt complex. So, this semi solidified polymer salt complex is then added to the acidic aqueous solution or to hot water. So, what happens when you do this is the ammonia and carbon dioxide gets generated and starts effervescing, getting released into the water. So, from the salt polymer blend you now have the gas leaving, and so this leaves a porous scaffold. So, you end up with a macroporous scaffold which can then be freeze dried to actually be stored for further use. So, the scaffold which you have prepared using gas forming and salt leaching method would look something like this. So, what you see here are pores which have good interconnectivity. So, because the gas is actually pumped through the pores, you would have nice interconnectivity for the pores. So, you can see that there are lots of pores. You look at the SEM image. So, you see that there is a pore and these are some of the pores, and as you see most of these pores are very deep, and even in this particular pore you would see there are smaller pores which are much darker in color, indicating that they are much deeper and they are actually probably interconnected to some other pore. So, this kind of a highly interconnected pore can actually be very useful when you are talking about tissue engineering applications. So, as I had already mentioned, cells will be able to infiltrate, you would also be able to get blood vessels forming, and also it can help in good diffusion. In early stages of any tissue being implanted, nutrient supply has to come through diffusion. So, if you have such highly interconnected porous structures, so the diffusion will be effective, thereby it will not be a limiting factor when you are talking about tissue formation. Let us move on to the next strategy. So, one of the strategies which I mentioned is formation of microspheres. So, here we will be talking about one of the 3 or 4 different strategies which have been used, where microspheres have been used to prepare scaffolds. So, what is done first is the polymer is dissolved in a solvent, and then you start dropping it into a solution. First this polymer is dissolved, and then it is actually dropped through a small pore using maybe a syringe pump or something, and then it is being mixed, it is stirred, and then it is centrifuged to remove these beads, these microspheres, which are washed and then freeze dried. So, these freeze dried microspheres can form structures like this. So, as you see, these are nice spheres which are present, and you see the cells which have adhered on this as well. So, these images which you see on the scaffold are actually cells which have been adhered to the scaffolds. So, because you have these microspheres which have assembled to form the scaffold, you also have a porous structure which is present in between these spheres. So, this can help in infiltration of nutrients and also hopefully cell infiltration. Okay. So, this is what is done for preparing microspheres. Moving on to the next topic, you have freeze drying. So, freeze drying is one of the most commonly used methods for creating a scaffold which is highly porous. So, what you do here is you basically create the polymer solution, and you pour it in a mold, and this mold, this polymer blend is actually then frozen and kept for freeze drying. So, freeze drying is also called as lyophilization, and after freeze drying it is washed and dried. You can also re-lyophilize it to make sure that you have a completely functional scaffold. So, when you observe these scaffolds under a scanning electron microscope, it would look something like this. So, these are actually scaffolds which were prepared in my lab, where isabgol, one of the carbohydrate polymers, was actually lyophilized to form scaffolds. As you can see, it is a highly porous structure, and you have huge pores which are very nicely interconnected as well. So, this kind of a highly interconnected structure, which you can see in the magnified images which are highly porous, this kind of structure can help in cell infiltration and also with nutrient diffusion. So, this is one of the common strategies which is used. We will look at the principle which causes this kind of a porous structure. So, in the earlier cases where we looked at salt leaching or gas forming, it was quite simple. We had the salt crystals which are present, which got washed away, creating the pore. You had the gas forming salt, which then released the gas, which could be ammonia and carbon dioxide in the example we showed. So, this caused the pores. However, here where are the pores coming from? We only did lyophilization. For that reason, you need to understand what the process of lyophilization does. So, let us look at the phase diagram for water. So, this is a simple phase diagram where it is between the plot between pressure versus temperature for water. So, you have 3 phases, you have the solid phase, you have the liquid phase, and you have the gas phase. So, these 3 phases are existing in the given temperature and pressure conditions. This point is the triple point. So, the triple point is where you would have all 3 phases existing in equilibrium. So, you also have these curves which are the vapor liquid equilibrium. So, this would be the gas liquid equilibrium curve, and you would have the solid liquid equilibrium curve here and the gas solid equilibrium curve here. So, these are the curves which you have in this phase diagram. So, now when we are talking about lyophilization, what do we exactly do? See, when we prepare the polymer blend and we actually pour it into a mold, so you have liquid water. So, this liquid water, if it is evaporated, would be the process where you have liquid under some condition moving to gas. So, this is the process of evaporation. However, when you are talking about freeze drying, what you are actually doing is not evaporation. You are removing the water at very low pressures. So, what you do is you freeze the water to form ice crystals, and these ice crystals are directly, so these ice crystals are directly sublimated instead of being evaporated. So, it does not first it does not actually melt to form water and then get evaporated. Instead, these ice crystals directly sublimate to form water vapor. So, how is this accomplished? This is done because at very low pressures in this region where you see the gas solid equilibrium curve here, if you actually have the conditions where solid can move to gas under this condition, under these pressures, then you would end up with sublimation instead of evaporation. So, what is done is you actually cool the water so that it solidifies, and then you reduce the pressure to such a large extent that this solid ice crystal then becomes a gas. So, by creating a vacuum and reducing the pressure significantly, you can actually convert solid ice crystals into gas. So, this way you are actually creating what you would see with salt leaching or gas forming techniques. So, what you have done here is you have created ice crystals within the polymer blend, and this ice crystal is then sublimated. So, causing something like a gas forming effect. So, this results in formation of the highly porous structures. So, this is one of the most commonly used methods for forming scaffolds. So, let us move on to the next topic, which is electrospinning. As I already mentioned, some of the extracellular matrix is fibrous in nature. So, it is important to create a structure which mimics the natural ECM. So, for this reason, people have looked at some of the textile based techniques such as electrospinning for creating scaffolds. Electrospinning creates nanofibrous scaffolds which have been studied extensively for a variety of different applications. So, let us look at electrospinning here. So, what is electrospinning? Electrospinning is a technique where you apply a high voltage to create very thin fibers from a polymer solution. So, what you do is you create a polymer solution which is loaded to a syringe, and this syringe is pumped using a syringe pump, so that the polymer solution comes out of a needle. This polymer solution which comes out of the needle is exposed to a high voltage, and because of this high voltage it would actually form very thin fibers. These thin fibers are collected on a collector which could either be a flat surface or it could be a rolling drum and so on. So, here the parameters which can be varied are the flow rate at which the polymer solution is pumped. You can also vary the viscosity of the polymer solution, and you can vary the diameter of the needle. You could vary the voltage which is supplied. There are also parameters such as humidity which can play a role in how these fibers are formed, and based on the distance between the collector and the spinneret, you would also have the thickness varying. Another factor which can be looked at is the different types of collectors which are used. By using a flat collector, you would be able to get nanofibrous mat which is not aligned. However, if you were to use a rolling drum or something, you can actually create aligned fibers. So, the scaffold, when you look at under a scanning electron microscope, would look something like this. What you see on the left is non-aligned electrospun fibers, and what you see on the right is the aligned electrospun fibers. So, these are some of the commonly used techniques which are used for scaffold fabrication. So, we have given an overview. We have not gone into great detail of each of these techniques. Depending on the polymer which we have chosen and the application for which we are working on, we have to choose fabrication strategies which will provide scaffolds which mimic the natural ECM. With this, we will move on to some of the more advanced techniques which are being currently researched for scaffold fabrication, which would be self assembly and 3D bioprinting. So, self assembly will actually be discussed by Ramya, one of my students. She is currently working on her PhD. She will describe how self assembly can be used in tissue engineering. her research is focused on self assembly. So, she would be able to give a very nice perspective. See you all in the next lecture. Thank you.